So, Caitlin Hester, December 11th, 2019. For my case connections, I started out with case 2, and that was PKU. Just basically, PKU leads to an accumulation of phenylalanine to such levels in the body that they become harmful, and they often manifest with neurologic consequences. So basically what you would need in that infant is a phenylalanine restricted and often tyrosine increased diet. So the reason why you need that increase in tyrosine is because it has now become a newly essential amino acid. So we look at this as a conditional amino acid, and this has to do with the metabolism of phenylalanine via phenylalanine hydroxylase. So if you do have PKU, the issue lies in the phenylalanine hydroxylase enzyme, and you don't get that hydroxylation of that phenylalanine side chain to make tyrosine. So that's why it now becomes an essential amino acid, because the body can no longer synthesize it. Potential causes for the presentation of PKU. So you have large neutral amino acids, phenylalanine being one of them, known as LNAA. And those are carried to the brain by the LNAA transporter 1, so LAT1. And phenylalanine has a high affinity for the LAT1 transporter, and so due to such high phenylalanine blood levels, it saturates these LAT1 receptor sites and prevents the uptake of other large neutral amino acids. So this leads to decreased protein and neurotransmitter synthesis, which is some of those neurologic effects and detriments you start seeing. And this can manifest with failure to thrive, mental disability. And then also with this, the lack of tyrosine made also has negative effects. So tyrosine is needed for dopamine synthesis in the brain. So when you lack that tyrosine seen in PKU, you get a decrease in dopamine synthesis. And according to Klein, dopamine functions in neuromodulation such as motor control, motivation, reward, cognitive function, maternal and reproductive behaviors. Tyrosine is also responsible for catecholamine creation, so that's norepinephrine, epinephrine, thyroid hormones T3 and T4. So those are systemic issues that you will start seeing in hormone due to hormone imbalance. So another remedy for this is giving a slow release of tyrosine to avoid significant fluctuations of tyrosine throughout the day. So the last part of this case study is the tetrahydrobiopterin, or the BH4 test. So BH4 is a cofactor needed for the conversion of phenylalanine by phenylalanine hydroxylase to tyrosine. So BH4 works with phenylalanine hydroxylase. So this test helps to distinguish the reason why you have elevated levels of phenylalanine in the blood. So what happens is they give that BH4 cofactor dose, and if there's a significant drop in phenylalanine after the BH4 induction, and the tests are measured at 4 and 8 hours after, this implies the elevated phenylalanine is due to a BH4 deficiency. But if there's no real reduction in phenylalanine levels in the blood, this is likely due to a phenylalanine hydroxylase deficiency. So in this case, patient B showed the significant drop in phenylalanine levels after administration, so we are thinking of a BH4 deficiency in this case. Patient A, the original patient, does not have a drop in phenylalanine, and so we think this is most likely the type 1 or classic phenylketonuria, due to the fact that phenylalanine hydroxylase is most likely the deficient enzyme in this process. So for case 3, the patient presents with hyperuricemia according to his lab results. His uric acid levels in his blood are higher than the normal range, and he is under excreting uric acid in his urine. The normal range is 335 to 491, and his is about 215 milligrams in 24 hours. So initially you think, okay, under excretion of uric acid, that's the issue here. And we'd be partly right, but you'll see further in the video why that is not just the case. So you think choice 3, decreased excretion of uric acid, and this would be in line with the fact that most cases of hyperuricemia are due to a renal impairment leading to underexcretion of uric acid. And we get this just looking at the Western culture and diet, how it's so changed toward, and we have lots of comorbidities like hypertension, and renal disease and impairment is not something that is few and far between in our population now. But then we want to rule out overproduction. And in order to rule out this, you need to look for maybe they have a purine-rich diet, errors in purine metabolism. So errors in purine metabolism, you would want to look at pentose phosphate pathway overactivity. And so you would then be looking at getting a metabolite profile or enzyme assays. So this might be the disease called von Gierke, which is overproduction of ribose 5-phosphate, leading to an increased purine production. And purines are then broken down into uric acid, so that's how you get the hyperuricemia. Also states of constant cell breakdown turnover, so this is like hemolysis, rhabdo, tumor lysis, and myeloproliferative states. Again, that's a thorough workup and differential with biopsies, metabolic profiles, etc. And then also you would want to rule out a decreased salvage of protons, or purines, sorry. So this is often errors in purine recycling, and this needs DNA analysis before you can make that diagnosis. So one of the more common ones, common for an issue in purine salvage, but errors in purine salvage themselves are very rare. But one of the disorders is the hypoxanthine guanine phosphoribosyl transferase, or HPRT, and deficiency, and this is an enzyme involved in purine salvage. And so when this is deficient, you are no longer able to salvage these purines to nucleotides, and you also get a host of other neurological problems. So again, the accumulation of these purines leads to increased uric acid production and hyperuricemia. So as of now, we think it's just choice 3, but then we learn more about the patient's diet. And the patient states that he drinks a lot of, he has recently switched to the non-diet versions of sodas, and all the sodas that he drinks and the substantial amount that he drinks are high in fructose. So fructose metabolism depletes ATP shortly due to the generation of AMP. And the ingested fructose is phosphorylated to fructose 1-phosphate via hepatic fructokinase, and there's no negative feedback in this reaction. So ATP and phosphate levels quickly decrease, and then the decrease in phosphate leads to the AMP deaminase to catalyze that degradation of AMP to inosine monophosphate, which increases purine degradation, producing more uric acid. So the final answer is the patient's clinical presentation is a combination of decreased excretion of uric acid as well as an increased production of purines via increased fructose consumption. So any suspicion for further issues in purine salvage or metabolism or overproduction, would definitely need additional clinical analysis, and you would also want to look for additional clinical symptoms as well. So the recommendation would just be diet modifications and starting on a xanthine oxidase inhibitor like allopurinol, seeing if the symptoms reside, continuing to follow up with blood work, and then if there's no resolution or things are getting worse, further investigating down those more detailed clinical testing to see the issue with the symptoms. And all the references are attached in my script. Thank you.